2. Chromosome mutation. Any change in the number and structure of the chromosome or chromosome sets, leading to ploidy e.g., polyploidy, trisomy, and chromosomal aberrations e.g., translocation, respectively. For details see chapters 14 and 15 in Part E Genetics. O. Advantages of mutation theory. The advantages of de Vries's mutation theory are the following. 1. 6. The mutation theory describes the importance of mutation in selective value of organisms. The mutation theory explains the occurrence of evolutionary changes within short period in contrast to natural selection, which describes slow and continuous variations. Mutation theory explains the absence of connecting links is no criteria against evolution, but its possibility exists. Occurrence of mutations in large and divergent direction removes the possibility of species disappearance by crossing. Since mutations appear fully formed from the beginning, there is no difficulty in explaining the incipient stages in the development of organs. Mutation is of great service to breeders in developing new useful varieties. Objections to mutation theory. 1. 2. 3. The mutation theory was unable to explain the presence of flightless birds on oceanus lands. It could not explain the existence of discontinuity in distribution among individuals. Many mutations, described by de Vries in Oenothera lamarckiana, are now known to be due to certain numerical and structural changes in the chromosomes. For instance, Gigas, mutant of O. lamarckiana, was later found to be due to polyploidy. Mutation theory alone could not explain evolution. It, however, provided raw material for other forces to act upon it and bring about evolutionary changes. Epigamic selection by Pehan. Selection in action. Examples and types of natural selection. Darwin clearly saw the importance of selection as a D' evolutionary force in the natural world. He cites countless examples of adaptation through selection, and thousands of new cases are recognized each year. But one problem in discussing selection has usually been the correlation of experimental studies under controlled laboratory conditions with what actually happens in nature. During the last 40 years, a series of studies combining experiment and observation in the field have been carried out on the problem of industrial melanism in moths. They provide an exciting insight into the operation of selection under natural conditions. These brilliant investigations were undertaken originally by R. A. Fisher, 1929, and E. B. Ford, 1940, 1964, 1971, and more recently by H. B. D. Kettlewell, 1956, 1961, 1973, all of Britain. 1. Melanism in moths or industrial melanism. With increasing industrialization due to industrial revolution in Britain, entomologists began to record the replacement of light or gray colors in many different species of moths with dark or black ones, called melanics. A particularly good example is the peppered moth, Biston beetleria, where the dark form is referred to as carbonaria. The following facts were discovered. 1. The earliest records of dark forms, i.e., 1% in 1845 and 99% in 1895, came from sites near heavily industrialized areas, Manchester, Birmingham. 2. The highest frequency of dark forms was found in sites near industrial centers, hence, the phenomenon was described as industrial melanism. 3. Melanics usually occurred in night-flying species. The possible evolutionary explanation is that light forms are more conspicuous to predators, birds such as English robin, Orithacus rubicula, when they rest on sooty branches and tree trunks in industrial sites in the daytime. Under unpolluted conditions, light moths were almost mingled with lichen tree trunks and become well protected. Figure 6.1. There are, of course, possible alternative explanations such as that the melanism might be phenotypic, e.g., due to uptake of the industrial pollutants. However, breeding experiments established genotypic control and indicated that carbonaria trait usually segregated as if it was a dominant at a single locus. There are other possible explanations, e.g., a dark color might protect against the direct effects of pollution. To analyze the situation further, therefore, Professor H. B. D. Kettlewell, 1956, 1973, performed the following experiments. 1. To determine whether carbonaria, i.e., melanic moth, had a greater fitness than typical light-peppered moth, Kettlewell released marked individuals into both industrialized and rural areas. More marked melanic. Figure 6.1. Typical light-colored and carbonaria dark-colored forms of Biston beetleria moth on an unpolluted, lichen-covered tree after Hamilton, 1967. Forms were recovered in the industrial sites and more gray forms in the rural areas. 2. Direct observations were made on predation rates. Similar numbers of melanic and typical light moths were put on tree trunks in polluted and non-polluted areas. More melanics were eaten by birds in the rural areas and more non-melanics in the industrial areas. Obviously, natural selection in terms of bird predators plays an enormously significant part in industrial melanism. 3. One bird, the creeper, did not distinguish between two moths, i.e., melanics and gray. Figure 6.2. Typical light-colored and carbonaria dark-colored forms of Biston beetleria moth on blackened bark in a polluted region of England after Hamilton, 1967. Production. The act of selection in itself does not reveal the factors are. Kettlewell noticed that it feeds by creeping up and down the trunks of trees, and is, therefore, more apt to see the silhouette of the moth resting upon the bark of the tree than the color of the wings. Points numbers 2 and 3 are particularly powerful supports for the idea that melanism has arisen in response to predation pressure. Thus, one of the many impressive features of Kettlewell's studies lies in the clear-cut identification of the selecting agent. Selection, we may recall, has been defined as differential regencies that enable one genotype to leave more offsprings than another. We may demonstrate the existence of selection, yet not know the cause of that selection. We might have reasonably suspected that predatory birds were directly responsible for the differential success of the melanic forms in survival and reproduction, but Kettlewell's laboriously accumulated data provided that all-important ingredient, the proof. Lastly, gradual replacement of soft, high-sulfur, sooty coal to less smoky fuel or by oil and electricity, reduced the soot deposition on the trees. Conditions then became more suitable for the survival of gray moths, consequently their frequency once again increased. Bishop and Cook, 1975. 2. Australian rabbits. In 1859, a small colony of 24 wild rabbits, Oryctolagus cuniculus, was brought from Europe to an estate in Victoria in the southeastern corner of Australia. From such modest beginnings, the rabbits multiplied enormously and by 1928 had spread over the greater part of the Australian continent. According to an estimate, the number of adult rabbit was over 500 million in an area of about 1 million square miles. The rabbits caused extensive damage to sheep grazing pastures and to wheat crop fields. For controlling the population explosion of these prolific rabbits, the Australian government spent huge sums of money for many years. Trapping, rabbit-proof fencing, poisoning of water holes, and fumigation all proved to be largely inadequate. Then, beginning in 1950, outstanding success in reducing the rabbit population was achieved by a biological control method, i.e., inoculating rabbits with a virus that causes the fatal disease myxomatosis. The deadly myxoma virus was implanted into the tissues of rabbits in the southern area of Australia. In a remarkably short period of time, the virus had made its way, aided by insect carriers, mosquitoes, into most of the rabbit-infested areas of the continent. By 1953, more than 95% of the rabbit population in Australia had been eradicated. But, after their drastic decline in the early 1950s, the rabbit populations began to build up again. Evolutionary changes have occurred in both the pathogen, i.e., myxoma virus, and the host rabbit. Mutations conferring resistance to the myxoma virus have selectively accumulated in the rabbit populations. At the same time, the viruses themselves have undergone genetic changes. Less virulent strains of the virus have evolved. Frank J. Fenner, 1959. 3. Resistance of insects to pesticides. The evolution of resistance of insect species to pesticides has in recent decades been a fantastic evidence of the ability of living species to undergo genetic changes in response to challenges of the environment. C. Brown, 1967, Giorgio, 1972. The story is the same. When a new insecticide is introduced, a certain relatively small amount of concentration is sufficient to achieve a satisfactory control of the insect pest abundance. The necessary concentration gradually increases, until it becomes totally ineffective or economically impractical. In 1947, Sacca was the first to report that a population of the housefly, Musca domestica, had become resistant to DDT, dichlorodiphenyltrichloroethane. Since then, resistance to one or more insecticides has been recorded in at least 225 species of insects, including the malaria-carrying mosquitoes and typhus-carrying lice, and other arthropods. Usually resistance is specific to one or a group of chemically related insecticides, but occasionally instances of multiple resistance are reported. An interesting parallel is found in a mammalian species, the gray rat. A substance known under the trade name of warfarin was destroying rats, chiefly through its action on a blood anticoagulant and interference with vitamin K. Warfarin-resistant rats, mutant strains, appeared first in a